Assalamualaikum and a very good evening. So today I'm going to share my summary journal, which is Food for Thought, Developmental Neurotoxicity, the Challenges in the 21st Century and Intellectual Opportunities, written by Smirnova, Halbert Claes and also Hartung. So basically, I will start with the introduction first. Okay, the introduction states about the NT. What is the NT? So the NT, which is developmental neurotoxicity, it is tested the health effects of chemicals, which is nowadays only 150 substances that has been tested. So why only 150 substances? The first one, because the NT is very time consuming. It takes time to do the research and also to prove that the chemicals is very giving its toxicants effects to the human being. Only several compounds that have been established as the antitoxicants in man, such as methylmercury, lead, arsenic, PCBs, toluene, and also ethanol. This is written by the Krengen and Larrikin in the journal in 2006. Then they follow up to the second journal, which is in 2014. They stated they added six chemicals, which are manganese, fluoride, chlorpyrifos, dichlorodiphenyl trichloroethene, tetrachloroethylene, and also polybrominated diphenyl ethers. So the effects of these chemicals is very strong. The first one, because it is very vulnerable to the chemical perturbation especially in children, because their brain is still developing compared to adult. Okay, the second one, this is due to the immature of their blood or also the brain barrier. Also, because of the increased absorption versus the low body weight, and also it will diminish the ability of detoxify the exogenous chemicals. Okay, apart from that, even our CNS development itself is very a complex process, because it involves many different events such as cell differentiation, cell proliferation, cell migration, synaptogenesis, myelination, cell death, synthesis of neurotransmitter, and also the formation of receptors. Even each of these events, they has their own time frame. So, it will create a different window of vulnerability to xenobiotic exposure, which means the xenobiotic is not naturally produced in our body. So, the exposure is we expose to the chemicals. So, once our neurodevelopmental is already disturbed by these chemicals, there is only a little potential for it to repair, and it leads to permanent consequence. The permanent consequence is, for example, it will cause the lowered IQ, learning disabilities, and also autism and ADHD. Okay, the second one is it is very costly. The NT is very costly because in vivo experiment, first, it is time consuming. Second, it is very unsuitable for testing a large number of chemicals. Even one chemical, it takes about 3 months to be tested, and it uses approximately 1,000 red pups, which costs about $1.4 million. That's why the NT can be very costly. Okay, one chemical, it takes about 3 months to be tested, and it uses approximately 1,000 red pups to be tested. So the cost is about $1.4 million. Thus, the NT is very costly. Also, if the NT study is performed, the data can be very difficult to interpret, and it really contribute to regulation and risk assessment. Therefore, nowadays, the researchers, they use a battery of high throughput tests of modern toxicology to test the effects of the NT, to test the effects of chemicals. So this will help in facilitate and speed up the process of risk assessment and the identification of possible environmental, also the gene interaction future. Thus, many experts in academia, in government, and even in this industry, they have discussed prior to these problems on how to improve and speed up the anti-tests, and they also provide recommendations in the future so that the anti-tests can be improved. Okay, continue with consideration 1. In consideration 1, Smirnova et al. They has discussed on the epidemic of neurodevelopmental disorder. Okay, Ball et al. In 1994, they has stated that 1 over 6 children has been diagnosed with developmental disorder, and many cases that involve with our CNS, which is our central nervous system. This disorder of neurobehavioral development affect 10 to 15% of all births, according to Granger and Nurain in 2014. Examples of neurodevelopmental disorders are learning disabilities, neurodevelopmental delays, autism spectrum disorder, ASD, attention deficit, and hyperactivity disorder, ADHD. Okay, for learning disability, it affects 10% of children that attending public school. Meanwhile, for autism spectrum disorder, it affects 1 out of 110 individuals in US. For hyperactivity disorder, ADHD, it affects 14% of 4 million children born in US each year, according to Nurain 2012. Okay, due to all this, scientific evidence actually suggests that this incidence is actually increasing over the year. For example, the rates of autism has doubled over the last decade. This autism actually increase are not believed to be as a result of reclassification, but it might be due to the exposure to drugs and also to industrial chemicals. According to Fung et al. In 2001, 40% of prescription drugs have been withdrawn from the market because of its neurological adverse effects to human beings. So, the evidence of the contribution of drugs to autism has been proven by its exposure to early pregnancy, such as thalidomide, misoprostol, and also valproic acid. It is true that developing in CNS is more vulnerable compared in adult. Means the developing in CNS is more vulnerable to chemical compared in adult. So, according to Bonley and Campbell in 2005, they argue that actually the exposure to chemicals will cause a drop of IQ in 5 points, but actually this will not greatly affect an individual. But comparing to widespread exposure, for example to lead, it might could spread such deficit over the whole population. So it means that it will affect population very greatly. So according to Granger and Nurain in 2014, loss of cognitive skills actually reduce children academic and also economic advancement, and this actually they have substantial long-term economic effects on societies. So, for example, why it is stated that it will cause a long-term economic effects on societies? Okay, for example, if exposure to lead, it will cost $50 billion. Meanwhile, exposure to metal mercury, it will cost $5 billion. Meanwhile, in Europe, exposure to metal mercury, a loss of 600,000 IQ every year will cost a loss of 10 billion. So, meanwhile, in France, the lead exposure will cause annual cost loss of exceed around 20 billion. Therefore, IQ loss actually represent one aspect of developmental neurotoxicity, so the total cost will be very higher. So, however, the NT actually they stand out as prototypic emerging health effects. Together, this make the NT as a prototypic emerging health effects if the incidence is apparently increasing, so that there is public awareness. So a precautionary reflex increase suggests that increased testing and banning of possible heart rate. However, some people believe that actually this scenario with the emerging health effect actually giving a positive feed forward. Why? Because, for example, the research will increase, the funds will available, and also the bias of reporting positive will go spiral. So, only the costly political and economical decision can be made. So the balance between these two, between this economic and also the political, will assure a quality tool. Means they will give a best tools. So they also help to understand the extent of the threat and furthers the quality of tools to assess it. Okay, continue with consideration 2, epidemiological studies of neurodevelopmental disorders. What is epidemiological studies? Epidemiological studies is about studying the human under natural exposure. For example, like doing survey or collecting the data from the hospital. But researchers agree that this kind of study is not relevant to conduct because it is very costly and it took epidemiology some a very long time to be conducted. For example, it took some 50 years to show that smoking actually induces lung cancer. Okay, epidemiological studies is divided into two types. The first one is retrospective and the second one is prospective. The retrospective, the advantage is the cases or the disease are easily to define, but it is very biased in selecting a control group. However, in prospective, they do not have bias while selecting a control group, but it is very hard to define the cases or the disease, and it requires a very high number a high number of study to find a sufficient number of cases. For example, in babies and pregnant mom, it will limit the biomonitoring of exposure and the biological phenotyping of mother and child and their response to exposure. Okay, according to Rice, he noted about the lack of strategies for choice of tests, data analysis, and interpretation of results for neurodevelopmental epidemiological studies. Example, at ASD, there has been limited the date by the lack of prospective studies that include the prenatal window, ASD phenotypes, and simultaneous biosample. There are key problem in case of the anti. The first one, there are only several the anti-toxicants that we know, and the second one, the environmental epidemiology, the exposure metrics are extremely difficult. Next, is there any need for the anti studies to be conducted? Because most chemicals are never tested for the anti since there are no general requirements for the anti studies. NRC, US National Research Council, they estimated that 30% of developmental disabilities is due to the neurotoxic exposure plus the genetic susceptibility. Meanwhile, 25% is due to environmental exposure. When the anti-studies actually should be conducted? Okay, first, based on observation neurological effects or induced structural abnormalities of CNS, whether the chemical should be recognized to have a possible trigger or not. The second one is the chemical trigger can be adult neurotoxicants, hormonally active peptides, and amino acid, or the chemicals that structurally similar to other chemicals with the anti-effects. However, due to commonly used experimental designs or the data is not always available. For example, chemicals with widespread human exposure, such as to be tested for reproductive toxicity and adult neurotoxicity before making decision for further DNT studies. However, regulation mainly they require reproductive and developmental studies and for high production volume chemicals. Also, adult neurotoxicological studies are only demanded if triggers have been found. Currently, there are only 200 chemicals that has been performed by the anti-testing and only few that contribute to risk assessment. Why? Because the first one, the endpoint is not sensitive as the previous test, and the second one, the data was not complete or very difficult to interpret. Okay, as for the anti, what actually makes the anti different from other neurotoxicity? Actually, our brain is a very complex, and they have unmatched complexity. The development lasts until puberty, and they create a window of vulnerability beyond the neurotoxicity effects in adults. Okay, the specific vulnerability are very dose dependent. For example, the lower concentration can damage the developing nervous system. Like for a nicotinic agonist may show transient neurotoxicity in adults, but the effects are adaptable. However, during the development of our nervous system, appropriate signals are required for formation of correct networks, and if there is any disruption during this, there will be a permanent effect. Okay, much of these variations between the difference of adult and also between our adult brains and developmental nervous system is explained by the variations in exposure and pharmacokinetics. For example, okay, children, they drink more water, they eat more food, and they breathe more air per body weight compared with adults. Like in Larrican et al. 2004, he stated that children in the first 6 months of life drink 7 times, where children ages 1 through 5 years eat 3 to 4 times. So actually, children, they have substantially heavy exposure compared to adults. Moreover, their metabolic detoxification is still immature, and the blood-brain barrier is still not fully formed. So that's basically the difference between the children and the adult nervous system. Okay, now we talk about the current DNT studies and the limitations. So currently, many widely used chemicals were never sufficiently assessed for DNT testing, means they are not assessed for human and environmental safety. Means even the DNT, they're still not fully required by most agencies, and it is only a must when they are triggered. The chemical used with different regulatory such as the pesticides, insecticides, food additives, and cosmetics. They always have, and they don't have to undergo the same testing. Next, however, since children are considered as a susceptible population, they require more extensive evaluation, and US EPA, they recommend that the anti studies for food use pesticides should be done for the children. So currently, ECD has published a reproductive toxicity test guideline that allow to assess the impact of chemical exposure on developing nervous system. The aspects that evaluated are the life stages, prenatal and postnatal, up to puberty, and the impact of development up to adult. So, in Europe, REACH regulation means the registration, evaluation, authorization, and restriction of chemicals. They went to force in June 2017 because they has their own aim. What is the aim? The aim is to improve the protection of human health and environment by how? By provide a better and earlier identification of toxic properties of chemicals. This includes a systematic evaluation of chemicals that will be produced in significant quantities. All producers and importers register the chemicals that produce in volume that greater in 1 ton per year. 1 ton per year at ECHA, which is European Chemicals Agency. And this includes this includes information of properties, users' risks, and the safest of handling the chemicals. The chemicals of very high concern, such as bioaccumulative, carcinogenic, mutagenic, and reproductive toxic compounds, is required specific authorization before usage. Mean these chemicals that causing unmanageable risk will be phased out. Mean it will be banned or partial or total ban from the market or the company. So, in the REACH testing scheme, there is no requirement for the anti-testing, and currently to increase the anti-testing requirement, it is in case of any noticeable neurobehavioral changes observed during systemic toxicity evaluation, they recommend that the testing for neurotoxicity or the NT according to the OECD guidelines. So everything that must be done must be followed the OECD guidelines. Okay. And currently, there are several endocrine disruptors that have been identified as the antitoxicants. The NT guideline is based on, and the current NT guideline is based on in vivo animals. Means the aim of the observation is to detect the neurological and behavioral abnormalities. Okay, this is the fact that estimated 1,000 rat pups is used for one NT, and imagine to produce these 1,000 rat pups, it requires 140 mated females needed to produce this enough pups. So it is surely that the NT it is like very costly because it requires that large amount of animal that be tested, and also the NT studies they took almost 3 months. Why? Because okay, out of these 1,000 pups, 640 are kept for at least 3 weeks, and the rest, which is 2,040 pups, they kept up to the young adult stage, which means 60 days postnatal. Together, if we sum up the postnatal and the prenatal period, approximately the NT studies they last for 3 months, so it is very time consuming, and they're also very costly, and it is very complex. The guideline for the animal to be tested, like how the way the experiment conducted, is very complex and complicated. Okay, currently OECD TG46 being the test guideline 426, they use rodents that treated during the gestation and also the lactations. Means this process it covers the prenatal and postnatal period of nervous system development. In postnatal, a core battery of tests was established to detect the disorders such as the functional, the behavioral, and neuroanatomical abnormalities. And there are 3 recommended endpoints such as neurobehavioral testing, neuropathology, and pharmacokinetics. And this assessment they encompass physical development, behavioral ontogeny, motor activity, motor and sensory function, learning and memory, brain morphometry, and neuropathology. However, for learning and memory assessment, the methodology is very flexible. Next, as stated, the NT guidelines are very complex. They are very costly. They are very time-consuming, and it is not suitable for routine testing of high number of chemicals. Although the protocol of the guidelines is well designed and it covers a broad of window of exposure, but there are problem arise such as the critical phase for some effects might be missed and not all effects would be found. And the second one, the interpretation of result is difficult because of the knowledge gaps. Means, okay, we have to consider about our normal brain development on functional, structural, and molecule levels. So all the process must be considered. So it will be sure that the process will be very complicated. Okay, moreover, there are also scientific concerns regarding the relevance of the studies for our health effects. Means, okay, for example, if the testing can be performed only at high doses, which are not relevant for human exposure scenarios, means if human routine, they are exposed to the low doses of chemicals, but in a very long time of periods. But it doesn't actually represent actually the experiment that conducted, actually it doesn't represent our life scenario as human. So the animal tests, they cannot reflect our individual uh, genetic differences. Okay, however, there are also scientific concerns regarding the relevance of the studies for human health effects. Means the testing only can be performed at high doses which are not relevant for human exposure scenarios. Okay, for example, we as human beings, we are exposed to low doses of chemicals, but in a very longer period. Means it is low doses every day, but it takes a long time, long period. But compared to the experiment that conducted, of course it will be, of course the chemicals perform at high doses, but in a very shorter period. So it doesn't actually represent the human exposure scenarios. And the second one, the animal tests also cannot reflect our individual genetic differences. And also, the interpretation of the behavioral effects and histology of in vivo data generated can be very difficult and less quantitative, which make it hard to predict human health effects. And the study design is of course very demanding, and the offer resulting in compromised study reports. Okay, starting in 2005, a group of experts in this kind of field, they have organized a workshop or conference so that they are together gathering to search or to discuss about other alternative for the anti-assessment and to provide recommendation in the future. The next step of the anti is the strategy is based on to increase the understanding of mechanism. So far, the toxicity pathway that has been identified is including the cell proliferation, precursor cell differentiation, cell migration, glial reactivity, glial maturation, and also those migration, apoptosis, synapse formation, synapse pruning, neurotransmitter receptor profiles, and neuronal connectivity. Somehow, this actually offers the opportunity to guide the future research. Okay, so next, what should we consider in order to bring forward effective and successful in vitro DNT testing methods? Currently, it is believed that in vitro methods can be used to conduct neurotoxicity testing because it can provide better understanding of cellular and molecular mechanism that involve in the anti. So, how we can make use of in vitro DNT tests to support epidemiology? There are several alternatives. Okay, the first one is the simple two-dimensional means study cell lines to more complex primary cells stem cells, 3D cell culture, and non-mammalian organisms. However, the NT is ultimately the consequence of disturbance of relative basic biological process, such as the differentiation, proliferation, migration, and new growth. Therefore, several systems in vitro that has been established which test the disturbance of such biological activities by chemicals. Next, apparent predictive the apparent predictive of zebrafish as a model for the anti-screening that it shows a very high correlation animal model as well as with human data. It appears that the test can be performed efficiently, reproducibly, and reliably at an early life stage and offer the possibility to combine assays at biochemical, cellular, and molecular levels with observation. Means zebrafish it is used as the anti-model because they has a very like the similarities towards with human our with with our data, so the tests can be performed efficiently, reproducibly, and reliably. So although most of these embryological process and molecular pathway in development have been conserved between zebrafish and mammalian species, there are still questions that remains, such as is it the model is predictive for human response? Since we have to consider about pharmacokinetics, which is the movement of our drugs in the body. Since the pathway across is like it's kind of different, so there are several things that still can be considered. So there are various number of challenges that exist in vitro DNA testing. Okay, the first one is the appropriate dosing, means the dose. Okay, appropriate dosing it consider that the dose at appropriate measure, means it doesn't should be too low or too high, and it is important to consider the measure because most cases the nominal concentration is the most practical approach, but sometimes there are highly hydrophobic compounds such as metal mercury. The cell actually the cell dose can be much higher than the expected normal. Means we have to make sure that the chosen concentration will not causing mere cytotoxicity. Cytotoxicity. Okay, means if we the choice of nominal concentration will result in specific effects. So the next thing is we have to make sure if we choose non-cytotoxic concentration, it actually has advantage due to the simplicity of the rules, but there are also a problem because cytotoxicity can depend on small details of the experimental system, and all of this they can if affect the final the anti-test concentration. However, if the meaningless high concentrations tested if a compound has a low toxicity. Okay, meaning of this like this. In the studies, the measurement too far into means if the measurement of the cytotoxicity is very high, it can or it is the measurement too far from the cytotoxic range, it can cause incorrect result. It can real incorrect results. But if the measurement at a very low concentration, it can reduce the power of the study. So that's why we have to make sure that choosing the best concentration we have to consider to choose the best concentration, because we don't want to make it like too high or too low. So um, this in such cases, they are suggested that anchoring the physiological concentration would make more sense. However, this is often prohibitively expensive for some endpoints. Means if we perform in a very large screen, they are actually very expensive for some endpoints. Things that should be considered is the role of exposure timing. Exposure timing, it should depend on the endpoint use, but there are still rules that need to be followed, or otherwise the data can be very difficult to interpret, or they require a lot of background information for interpretation. The consideration is, okay, if it is a short exposure, it will give information on the direct signaling changes. Means the short exposure are more likely to give information based on the direct based on the it will give information on direct signaling based on the changes that triggered by the chemicals. And also, it must be noted, if the test system is show different response at different times, may show different responses at different times, but for long exposure, it will yield many of the informations on the adaptation to the systems to the toxic challenge. Okay, so basically the timing considering the timing of exposure is very important, because um, the sensitivity of differentiation stem cell is different. Okay, for example, exposure of human stem cell during the first 4 days of differentiation is completely will alter the differentiation track. Meanwhile, the exposure from day to 4-6 on day 4 from day 4 to 6 or from day 4 to 10 actually had no effect. So we can see best the effect of exposure there with the times, with the difference of time. Okay. Next, how to deal with the time offset. Okay, our nervous system is issue the issue of our nervous system plasticity is linked to the question of the time offset. Like, the nervous system can adapt to damage, and it can show a very high plasticity that can make up for effects damage over time. For in vitro models, this means that the toxicity can be missed in cases when increased proliferation leads to similar final cell numbers as in control conditions. Actually, an important mechanistic that an important things that deals with our memory damage is, how does the system know that actually it has been disturbed by the chemicals earlier. Okay, for example, if cell migration is disturbed during critical phase, the cells will end up in wrong place, and it will delay the DNT effects. But in other cases, they may be molecular correlates of damage memory. So we actually don't know if either the chemicals is actually actually the factor of the anti effects, or actually it is because of other cases that correlates with our molecular correlates with our damage memory. Okay, the next consideration is how actually the adversity can be defined in experimental system. The question is, how do the researchers can differentiate or distinguish between the toxicology effect and the significant effect, because our body or our living system will respond to a stressor, means to a toxicants, to make sure the hazard is less severe or less serious. So the response, they might be in exploratory or like sensing the impact of the stressor or mobilizing the defense pathway. So this will lead to overestimation of adversity. The second one is how the second question that always be asked is how the adverse effect can be ranked or be quantified. Be, because okay, this is also one of unsolved questions because now even the studies there is only a few that attempted to rank the compound by develop a DNT toxicity index. Means to develop only few studies that attempted to rank the compound by potency or to develop a DNT toxicity index. And there are also questions asked that what is the constituents and adverse effect as a point of reference for validating the anti-test. Means, for example, we cannot say that um, alteration in MRI scanning studies on altered human brain responses represent adversity. So we don't know which actually should be the reference of how do we know it is a very adverse or it is only less severe. The effects is less severe or very severe. So we actually there is no point of reference to validate the anti-test. So um, we can say that this question that arises, there's still the researchers still considering about all this since there are no like how do we say that? There is no proof or there is no answers. There's still no answers to these questions. Okay, the next consideration is how the DNT will be related to cytotoxicity and the organ-specific toxicity. Okay, general cytotoxicity is served as estimate of neurotoxicity and can actually this neurotoxicity can be estimated from other organ toxicity. Okay, the next consideration is how the DNT related to cytotoxicity and the organ-specific toxicity. Okay, for in vivo toxicology data, the interpretation of studies and the extrapolation to man is a potential DNT observation, a primary effect of chemical exposure. Thus, it will be potentially have relevance to man, or secondary effect of other toxicities may not occur in man. Okay, a simple parameter is to know whether the DNT effects are observed in the absence of maternal toxicity. So like if these cases mean the data is easy and clean to interpret, but if the DNT is observed at doses that lead to maternal toxicity, means the data is complicated. So is this like it is hard to interpret. Okay, for examples, the first one is there are many forms of maternal toxicity. The first, for example, hepatotoxicity and also pulmonary toxicity. Hepatotoxicity is um, related to our liver, meanwhile the pulmonary toxicity is toxicity that related to our lung. So this will lead to general wasting and weight loss in the fetus, such as like associated with misbalance of nutrients and vitamins in the blood. So this will lead to secondary development defects, DNT, which is low relevance to man. So if the such if the like the situations, it is not easy to judge. Either they are more than 3 doses. It is sorry. In this situation is not easy to judge as typically it is not more than 3 doses. So the it may occur that one is too low for the anti-effect, and the next higher doses will show the anti-effects and metallotoxicity. So the next example is warfarin. They can show the anti-effects in animals by how? Okay, the compound in the warfarin in the compound is a vitamin K vitamin K antagonist, and basically it will affect our blood clotting factor in the liver. So it will lead to internal bleeding. So the indirect this is for example of indirect how the indirect DNT occurs. However, there are also some vitamin K dependent enzymes that could be directly involved in our system development. So the data still insufficient for this model toxicant to resolve the issue, since it is stated that there are some vitamin that K dependent and there are some vitamin that antagonists. So the researchers cannot distinguish whether the model toxicant whether the data is valid or not. The third one is methyl azoxymethanol or MAM. Okay, this this it has a very simple straightforward mechanism action. So for in vivo studies, MAM um, it will illustrate several it will illustrate how actually the DNT occur first. MAM the would be maternally toxic in a standard dosing, means they has a standard dose to be to achieve the level of toxicant. So it if the it will in standard regulatory studies most likely not be toxicants, not mostly not classified as the antitoxicants. Thus, it can be used in a single dose, means to kill only a specific neuronal precursor subpopulation in the embryo. But also this result in neuropsychiatric phenotype with other types of toxicity. So a simple cytotoxicant like this, they somehow can resulting to be the anti-compound. And uh, okay, and the last example is thyroid toxicants such as metimazole and thiorease. These two, they can be very highly organ specific. They can destroy the thyroid without giving any significant effects on the nervous system. But this thyroid hormone, they literally play an important role in formation of our nervous system. So thyroid toxicants is the best known as establish the anti-compounds, even though the effects are only indirect. So these four examples, they gives a good indication of what should we consider and when selecting test compounds for in vitro studies and to interpret data from in vitro testing. Okay, the next thing that should be considered is the immune contribution to the anti and its testing. Okay, so currently there are so many well-established immunotoxicants that known to be the antitoxicants such as lead, mercury, benzene, PCBs, arsenics, and so on. So this when this inflammatory and immune tests should be considered. So the next thing that should be considered is the gene or environment interactions in the anti. Why? Okay, currently there are many neurodevelopmental disorder, including ASD, which is the autism disorder. They don't have a clear genetic basis, so they sometimes appear partially due to gene or the environment interactions. So, the effects of the genetic background they cannot be tested using the normal testing such as using the animal testing. So a new way has been established to test this kind of things such as by the using of induced pluripotent stem cells iPSC. So, by using this iPSC from patients with neurodevelopmental disorders, um, it will be possible to test substance sensitivity in the same model with different genetic background, and it will answer that whether this particular actually they are from the genetic makeup or the person itself they are sensitive to the they are sensitive to the gene or the environmental stress. So, using this in vitro models based on iPSC will determine whether the gene or environment interaction did, uh, is that really they take place on the development of the certain disorders. So, um, also this IPSC they have the another advantage which is the differentiation in vitro is quite similar to the stage of our brain development in utero. So each of these stage they exactly the same like exactly the stage of the brain development is exactly the same, so there will be no problem at there. Okay. I'll move on to the next thing that should be considered is there also the anti for the peripheral nervous system, because most of the anti studies they're related or they're focusing on our central nervous system. Okay. There is an example. Our palate is a typical neural crest-related development disorder, but they are still not classifically as the anti-effect. While the neural crest cells they also involved in the proper closure of the neural tube, and the failure of this process can result in hydrocephalus or spina bifida, but they're also not counted as the anti-effects. And the next one is our neural crash developmental toxicity have been investigated as investigated in different model organism, but the human data they are very due to the limited availability of fetal tissue and the neural crest cells. So this due to this there has been used um, for example they have been used for establishing functional DNT and for the generation of peripheral neurons that may be used for the further test system. So by that there will be strong evidence that actually the chemical can affect the neuron's growth of peripheral sensory neurons. So apart from that, the researchers they also have to deal with species differences in all the species in the model system, because we know that when dealing with all these model organism, they has their own differences, species differences. So the researchers have to deal with these things, and also they have to deal with the quality control and the validation for the anti screening, since we know that the quality control is not unique to the anti, but somehow it is still worth that we have to consider in vitro approach to the anti. So, in conclusion, we know that this currently toxicological studies, which is animal studies, it is very hard to do. It is very complicated process because it is very costly, and the second, it is very time consuming, and the chemicals, they can be tested in a larger number at one time. So somehow this problem they lead to resulting to giving incorrect information or incorrect results. So the toxicologists, they have been aware of this limitation of the testing, of the current testing. So they are thinking what should they do for the testing to be advanced, since it is we are currently in 21st century. So there are many emerging technologies that been demonstrated for the development of modern approach for toxicology to replace the traditional animal based testing. So like for example, here it is stated that the biotech revolution, such as the organotypic cultures, stem cells, and more high content methods like the recent development, they are using 3D cell culturing materials, which is the advance in knowledge on stem cell differentiation allow the development of complex tissue structure. So um, this 3D cell culture improve the physiological relevance of cell-based and the comparability between in vitro culture and living organism. So like this 3D culture, they are more advanced because they will advancing the understanding of the molecular and the cellular mechanisms that um, actually occurred in the toxicity itself. And so basically like you know that this traditional testing, it is still, most companies they're still using, I mean like most companies they're still using this kind of testing, because only that we have currently the researchers that develop only that kind of testing that we develop. And mostly even though it is very hard, it is very complicated, but what can we do actually? That's the thing that only we have right now. So that's why there are some toxicologists, they want to be they want like to be advanced, since we are currently living in the modern world. So all the testing should be in a modern way. So um, hopefully like um the things like I don't know, hopefully like this what to say this development, this current development modern technology will develop well, so that it will be easy for the researchers to conduct more testing for the end for developmental neurotoxicology.